Hello, good afternoon, good morning. Uh, Very welcome to everyone joining this webinar, which will be covering emergency epidemiology. Myself and my colleague, past colleague from Towers Watson, but present colleague within the COVID ARG Actuaries Response Group, will be talking today about how emergency epidemiology has received a boost in the arm because of the COVID pandemic, and we'll be going through a number of different topics. So let me just share my screen. Hopefully everyone's able to see that. So here we stand in the middle of a 2 weeks of various different webinars covering different aspects of COVID and the pandemic impact on society and individuals. And today, wanted to cover a number of topics relating to the epidemiological side of the pandemic, and starting looking at some of the kind of early developments in epidemiology that's led to us this point, looking at data, the mutations and variants that we have heard so much about, and then Matthew Edwards will be kind of covering the precautionary principle and the way in which these variations we see, how we deal with that in the ongoing battle against COVID. So let me start by these three seemingly unconnected companies, Southwark and Vauxhall Water Company, Lambeth Water Company, and Metropolitan Life in the US. And what do they have to do with epidemiology? What do they have to do with where we are today? And the two at the top, really, the comparison between those two companies allowed John Snow, not of Winterfell, rather of London, John Snow to spend an enormous amount of time, and I think that's something I want to bring across right at the beginning, which is epidemiology used to be an incredibly large amount of work, trudging the streets, filling out surveys, filling out questionnaires, gaining information. And indeed, he had to have several attempts before his colleagues appreciated that what he was discovering, namely the distribution of cholera related to water coming into houses at standpipes, that actually this evidence base took an enormous amount of effort to put together and to gather. And Metropolitan Life, about 60 years later, actually they made a grant to the state of Massachusetts to look at tuberculosis, to set up the first ever community-based evaluation of tuberculosis, with people being tested and effectively diagnosed. And the success of that grant, of that study, meant that Framingham in particular became the site when they were looking for another source of community information, led to the famous Framingham Heart Study. And here we have many of the different continuing researchers that have looked at this and the underlying understanding and data relating to hypertension. But the important piece in all this is huge amounts of data needed to be collected, often by hand, often over many years. And that is very challenging in a time of crisis. And so this is typically one of the data that we might want to be using, that's gathered in terms of medical records, thousands and thousands, millions and millions of these type of paper records. And what's happened very, very recently is the increasing amounts of this data not only being digitized, but available. And here, this is a screenshot from my phone. And this enables me to look at my GP health record and enables me to kind of make appointments, but more importantly, to see the underlying data. And what's available to me, if you aggregate it together, if you de-anonymize it, is available to researchers. And during the course of the COVID pandemic, the increasing amounts of data being linked together in the UK through the NHS number, this unique identifier, means that primary care, secondary care, and indeed the variety of different tests, and we'll be talking more about those tests, can be brought together in one place and made available in a suitable format for approved researchers. So instead of trudging the streets as John Snow had to do, this information is available here at your screen, at your desk. And it covers the full totality of the healthcare experience, going all the way up to ICNARC with the intensive care units that we've heard so much about. So more and more data being brought together in one place. And one particular nexus of data that really only started a few weeks after the COVID pandemic started was Open Safely. And Open Safely brings together a number of different highly reputable research institutions and a very large underlying electronic health records data set, namely from TPP. And this data set of roughly 17 million people enabled the researchers to do a number of things. And here on the left, you can see analysis which is looking at mortality risks for different groups. And this is a mortality risk allowing for the interactions between different possible causative factors. And as we know, age is highly important, sex, and going through, we can see the relative importance based on an enormous data set which was constructed during the beginning of the COVID pandemic. And as time has gone on, this data set has increasingly been used to actually look at other things, and in particular, looking at vaccination rates. And here we can see the vaccination rates for one of the priority groups in the UK, those who have needed a shield because of prior conditions between ages 16 and 69, one of the earlier groups. And you can see over time, and this data is also available by geography, you can see over time how the rollout of that vaccination has progressed, and indeed where more focus is needed, where greater understanding as to why there may be different reasons that people are unwilling or unable to be vaccinated. And that needs to be addressed, because it's only through getting to high levels of vaccination with a disease, with a virus such as SARS-CoV-2, that you're able to achieve to get that level of herd immunity. And that's something we'll talk about more later as well. But something different has also happened, which is we're not just dependent on electronic health records, and we're not just dependent on trudging the streets. We now have, every one of us, or almost every one of us, has a smartphone. And since the beginning of the pandemic, smartphones have become an increasingly important tool in the epidemiologist's toolkit. And one in particular, which was put together by King's College London and the health company Zoe, the COVID symptom study, has seen an enormous amount of data being collected by individuals. And as of now, you can see roughly 4.5 million people have contributed. And what are they contributing? Well, they're contributing initially symptoms, a report on a day-by-day basis as to what symptoms they were feeling, whether they had COVID or not. And that information, that huge morass of information, enabled this type of information to be gathered and insights to be drawn. And in particular, the shift from the original focus on 3 symptoms to perhaps up to 35 different symptoms that might be linked with COVID-19. And that huge amount of data allowed King's College London to start to construct a kind of, from the symptom side, particular clusters to identify individuals that may or may not have a greater likelihood of needing particular support, respiratory support in particular. And also the ability to identify where it's likely that people were infected. And so if you go online, you can see any day, you can see the numbers of people that are estimated to have been infected. And you can see how that changes over time. And then that can fit together with the data we're collecting directly from tests, whether it be PCR or lateral flow tests. So a huge amount of data contributed by the individual. Citizen science is the phrase that's often been used. And this is a transformation from the old world of epidemiology, where it was driven by the epidemiologist. And the type of information that we gathered is incredibly valuable and useful. And one particular area was looking at the type of symptoms we see after vaccination. And here you can see on the left the symptoms recorded after people had taken the first AstraZeneca vaccine, with a high proportion of people reporting things like headache and fatigue and other potential symptoms. And if we compare that to the information that was gathered from the BioNTech Pfizer vaccine, we can see generally lower levels. But we also see an interesting pattern, which is that with the Pfizer vaccine, the effects, the symptoms seem to be higher, seem to be more widely experienced after the second dose rather than the first dose. So all this information is gathered by individuals. Therefore, there is this element, who records, who's more likely to use it, but it's a valuable data set for us to be able to look at. And so valuable indeed that actually the King's College London and Zoe group together tried to put together clusters of individuals, clusters of symptoms, which they believed were more predictive of particular eventualities needing for more serious care. And this graph is highly complicated in terms of what it's trying to tell you, but actually underlying it, probably two key things just to bring out. One is on the top, we're looking here, the more red something is, the more in excess of average likelihood of those symptoms occurring. And you can see, those of you with better eyesight than me, you can see the variety of different symptoms that are being picked up: skipped meals, loss of appetite, loss of smell, as well as the classic 3 that we knew from the beginning. And then the bottom panel, that is showing how likely or how significant is this particular result, an area that Matthew will be picking up later on. And the more red it is, the more significant that it is in excess of that which should be expected. And if we then track those clusters through to what actually happened to them in terms of needing care in hospital, the Zoe group was able to come with this conclusion, which was a range from cluster 1, where 1.5% of that population needed respiratory support, all the way up to cluster 6, where 19.8% required respiratory support. This is all information gathered through people's phone on their symptoms, which is providing information as to how best to treat or when best to intervene. And viewed a different way, perhaps a little bit more visible, then on the bottom we have the clusters here, 1 through 6, we have the various different symptoms, and on the left we have the percentage recurrence, and on the right we have the likelihood. And so you can see that when you get to the higher clusters, areas such as shortness of breath or confusion were highly prevalent in cluster 6, whereas, for example, whether you skipped meals seemed to be not important in cluster 1, but somewhat more important in cluster 2. So this type of analysis, this data, being able to pore over it, is highly valuable. But obviously we have many sources of data coming through to us, and one of those that people will be intimately aware of is the numbers of tests, whether they be PCR, whether they be lateral flow. And this gives you an indication of quite how many tests are being carried out around the world. And it's an enormous variation, everywhere from somewhere like Australia, where we're in the thousands of tests per individual case, to other countries where it's much, much lower, close to 0. And in part, that reflects the kind of the ethos of the underlying country in terms of how testing should be provided. In part, it also reflects the relative risk. But over time, country after country has been building up their testing capabilities. And that testing capabilities is linked to tracing. And going back to John Snow, rather than tracing around the streets of London, now we obviously have the ability through smartphones, through call centers, to be able to trace individuals. And it's the important piece is how widely do you trace? How many contacts do you trace? And so various apps indicate whether it's likely that someone may have been infected because they happen to be in a location where someone else has already proved to be positive. But this requires an enormous amount of effort, time, and people, and very clear workflows. And what's been fascinating is that this has built up to an enormous industry of testing. And here in the UK, we have a number of different pillars in terms of the various different tests that are carried out. So whether that's testing which is linked to clinical need in hospitals, pillar 1, the wider testing of the population to determine how variants and indeed the original virus may be spreading, trying to understand how it is that antibodies have developed and what the prevalence is and how that fades over time, and national surveillance programs. So going back to the original concept of epidemiology, tracking people over time, trying to find those who are asymptomatic as symptomatic. But as you can see, we are talking here in the UK about literally not just millions of tests, but 150 million tests. So an astonishing amount of testing going on, which we really haven't seen anything like before, and the prospect going forward of more and more of this testing. And that has led to obviously a number of questions, particularly when it comes to lateral flow tests, which are increasingly forming more and more of those tests, which is, what does that test tell us and how do we act on it? It's quick and therefore it gives you some information, but have we got false positives? Do we have false negatives? Are people who actually have COVID circulating who otherwise wouldn't, emboldened by their negative test, or are there people who are being suggested that they may have it when in fact they don't? And you can see here the level of positivity in the UK. So far over this period of time, about 26 million tests carried out, roughly 0.1% positive. 1,000 tests for every positive. And of those positive tests, about 20%, when they use the gold standard with PCR, polymerase chain reaction, about 20% proved to be negative. It's a tricky process. It's a process which is carried out by you yourself in many cases, or a parent, or someone who may not be doing this the whole time. And therefore, at the same time that this lateral flow testing is now available to everyone, having had different levels, is now available to everyone across the country twice weekly, if wished, we have a number of problems. The first problem is, it is a difficult task to do properly and accurately. It can be quite difficult and time-consuming to record the information. If you have a positive test, are those positive tests always recorded? If you are positive, then you are expected to quarantine, and therefore there may be a number that are not reported. One of the things about the lateral flow test is it is more effective for symptomatic cases than asymptomatic cases, and therefore there's a probability that some will be missed. And as the numbers fall, thankfully, as the numbers of infections fall, then that proportion of false positives becomes more and more important. Set against this, lateral flow tests are hugely scalable, able to be carried out on a kit at home and at relatively low cost. So the ability to get back to a more normal society provided by lateral flow tests is hugely attractive, but we must be aware that this is something which is potentially got a number of issues to deal with. And why are we so interested in the testing? And why are we so interested in particularly picking up the virus? And that's because, and many of you will have direct experience of many hours looking at these various different mutations. So I'm just going to summarize a little bit what's going on here at the kind of deeper level, because I think that helps in terms of understanding where we're going, particularly when Matthew takes over and talks more about different parts of the presentation. But underlying this, RNA viruses, of which coronavirus is one, are not that perfect in terms of replicating. Indeed, that is one of the ways that they evolve successfully, by adjusting their DNA or RNA to escape defenses. And compared to other RNA viruses, coronavirus mutates a little bit less, but it is still mutating at a relatively constant rate over the period that we have been tracking it. What's important is those mutations sometimes do or do not lead to a change in an underlying amino acid, and that change sometimes leads to an advantage for the virus or a disadvantage. And this is a very competitive world. The world of virus is very competitive, so we only get to hear about the successful ones, the ones where it made a difference, or the ones where a slight mutation meant the virus was completely either stymied from progressing or was slower than the existing, that one never gets to the attention of anyone. But the ones where there's a difference which makes either transmission or longevity, that's what we hear about. And as we get through this process, we hear about these variants under investigation. When they get more serious, variants of concern, which have been gone under risk assessment, and eventually variants of high consequence. Now, so far, we haven't had any variants of high consequence, which is good. As we get into this part, and we're getting more into exploring these areas that people may have less familiarity with, please, I would remind that we do have the question and answer on your screen. So if you'd like to ask any questions, then we're very happy to take those as we go through the presentation, or alternatively to cover those at the end. But as we look at these different variants, one of the things that becomes apparent is that there are particular places on the RNA genome of the virus which seem to be more likely to turn into mutations that we hear about. And that isn't always necessarily the same around the world. And so this is a variety of different places within the genome where mutations have been found. And the genome of COVID is about 30,000 base pairs. So compared to your genome, it's tiny. But in terms of the impact of those mutations, that's what we've been hearing about. And that's what we see when we look at the different variants. Now, this is a wonderfully colorful graph that we get from NextStrain, which has, prior to COVID, been tracking genomic sequencing across the world. And each of those individual dots, and there are many, many dots that you can't see, each of those individual dots is one particular virus that had been sequenced. And this is a time axis. And as time goes on, two things happen. One, you get a series of branches coming off where there's new lines or new lineages. And the second is that we move up the page. And the reason we're moving up the page is we're moving further and further away from that original virus. There are more and more potential mutations, sometimes individually, sometimes multiple. And three that we've heard more and more about over the last few months are the 501Y, V1, V2, and V3. Why is it called 501Y? Simply at the place where you have your 501st amino acid, then basically you have this switch between what should be there and what is there now. And that switch leads to a change in the protein structure. And we'll come to that in a second. But what it's telling us is that these genomic sequences are being constantly analyzed and new mutations are being picked up. And this shows you where, because it's not just one, where the various different variants are associated with particular amino acids that have changed. And it gives you a sense of how big the genome is, how many amino acids are involved. And indeed, these big blocks here are the different proteins which underline the virus. Now, here, when I show you that, it looks very simple, and you kind of think, well, okay, so it's just a nice long line of amino acids. But as soon as you create it, as soon as you create that line of amino acid, they immediately fold up. They immediately fold up, and this, in gray, is what the actual, in this case, the spike protein, looks like under these three different variants. And up at the top, you can see what the spike protein is actually binding into, namely the receptor, the ACE2 receptor. And so, although maybe to first eye these look identical, they are slightly different. And because of the different amino acid, the way they fold is slightly different, and therefore the way they interact with the receptor and the way they interact with antibodies is different. Now, there is a challenge here, and the challenge goes back to epidemiology. And the challenge is that we can't clearly sequence every single individual virus. And therefore, we're trying to sequence an appropriate number. But the capacity to sequence is very different around the world. And so at the moment, largely because of the number of cases, some of the countries which have the highest sequencing are places like Iceland and Australia. In the UK, we're about 80 per thousand. But as you go down that list, you can see that the number that we are able to sequence compared to the number of cases that are out there comes lower and lower. And why is that important? Well, as the number becomes lower and lower, there's obviously a challenge that sample to be sequenced, how representative is it? How representative the mix of different variants that are circulating? And are we picking up variants which are potentially more threatening, more likely to spread? Now, this speed of spread is really, really important, because if you look here at what happened in the UK, in different regions of the UK, then you can see over time how the variants, the dominant variant has changed. This one in purple, the B.1.177, which is not the one, not the Kent one. Unfortunately, it has a rather similar name, but it's not the Kent one. That was one that originated in Spain and was brought to the UK. You can see how that dominated, but then it was completely taken over by the more recent Kent variant, the B.1.1.7. And that particular variant, which is a higher transmissibility, potentially higher impact, that has spread around the world. And you can see here, this slide here, and how a relatively short period of time, a matter of weeks, the B.1.1.7 in the US went from being a important but a minority to a dominant form. And that speed, we're seeing in India as well, with the B.1.617, how variants manage to outcompete the existing and therefore become the dominant form. And just because it's better transmitting, it does not necessarily mean it has a more severe impact. But we need to track it and we need to trace it. Now, there has been some interesting discussion about these different variants. Are they trending through convergent evolution? Are they trending to a super variant? And there does seem to be some commonality in terms of particular mutations that occur quite frequently. This E484K that we've heard about in the Brazilian variant, in the South African, and now actually also appearing in the variant that's circulating widely in India. But there isn't a kind of, this is not by design. There isn't a destination. This is all by accident. This is mutation. This is evolution. But there are particular variants that have been found that are further away from the starting point. And here's one in particular. And this one in particular, which was found in Angola, which is 34 amino acids away from the starting point, and 17 of those amino acids in this spike protein. And you can see the variety. So this will continue to be a battle because of the variation, and a battle in terms of the vaccine. And as over time, those vaccines will get better and better at trying to attack the newer variants, but it is undoubtedly a battle, and a battle that continues to need to be won. And so with that, I'm going to hand over to Matthew, who's going to go a little bit more into the depth of what we do in the face of that battle.
Excellent. Thanks very much indeed, Dan. It's been a very hard act to follow. In particular, I don't have any super polychromatic illustrations on my slides, but anyway. But just to note to the audience again, and thanks very much for joining from the many different countries we're told you're joining from. You got this sort of Q&A box at the bottom of your screen, so if you want to start typing questions, you're very welcome to do so, and Dan, you're very welcome to answer those as we go along without waiting to the end if you'd like to. So, I mean, the advance of epidemiology over the last year has been quite astonishing, just sort of grabbing the opportunities it has through data collection, through smartphones and so on. But clearly a key thing in this new type of epidemiology, as well as, if you like, the older type, is all about decision making. I mean, what do you do with the analysis you've got. And one thing I like to do throughout the next few slides, also try and sort of draw parallels where possible with applications in your own, if you like, day-to-day insurance or other aspects of actuarial work in your day-to-day work. Because a key thing of analysis in general is there's no point doing analytics or any form of analysis unless you're actually going to use that to improve your decision making. So the key thing is, how does all this amazing research and amazing analysis that Dan's just been describing, how does it improve our decision making meaning obviously society as a collective whole in the face of the coronavirus enemy. So although in the agenda, in the description of the talk, I was using the phrase precautionary principle, effectively it's all about decision making. What is the thought process behind that decision making, which largely boils down to this aspect of precaution. How precautious should we be, and should that change in time of emergency as opposed to, if you like, the normal situation. So the precautionary principle, well, what's that all about? I think many of you will be familiar with it just from your own training in regards to significance testing. In other words, if you sort of cast your mind back either to your stats actuarial exams or to some of the more recent work you may have been doing, depending on what type of work you do, of course, you'll come along and you'll look at a particular sort of new factor in an analysis, and the default option is to assume that anything new is to be disregarded unless you've got strong statistical evidence that it should be included as a sort of important factor. And this is surprising. Maybe it surprises some of you, but it's actually less of a new thing than you might expect. It's less than 100 years old, this approach. So good old Fisher, it's always good having a chance to plug him. R.A. Fisher suggested this approach back in 1925 in the book I cite there. And at the time he was he's not particularly wedded to a particular criterion. He's mentioned 5% quite a few times. He's quite a few of his results throughout the book. We're looking at 10%, 5%, I think 2% as well, and 1%. But 5% seems to have become this of a bit of a default option ever since then, without there ever having really been any sort of particularly strong debate about it. But the key thing here, though, is that we're starting with the a priori assumption that the status quo, if you like, is correct, and anything new needs to be sort of proved to be worthwhile if we're to sort of take any notice of it. But the key thing here, though, of course, is that we might actually start from a different position, and we might actually assume that we have other perspectives, other evidence. So the example I often use is that of mortality on large data sets. If we start with the position that we know from all the evidence we've seen about pretty much everything in the field of mortality that male and female mortality is generally different in almost every single possible respect, apart from the fact that they both ultimately 10 to 1, we might look at a new factor and we might almost assume from the start that it's actually going to be different for males and females, rather than the typical statistical perspective of assuming it's going to be identical for males and females and looking for strong evidence that it's different. So again, it's effectively it's up to the designer of the analysis or the interpreter of the analysis to actually work out what the sort of your overall context is. So just to note that the way these tests are designed, it depends very much on where you think the starting point is. And of course, I've sort of obligatory in these things to have sort of one of the nice XKCD cartoons. So this is one of the good ones on significance testing. So the XKCD website is always good if you're looking to spice up your presentations. That's probably it for this slide, Dan, if you want to move on, thanks. So how does this all work in epidemiology? Well, the standard principle of medicine, what's hopefully a standard principle of medicine, is this idea of first do no harm, which is attributed to Hippocrates. I don't think it's actually part of the Hippocratic Oath, but it's generally seen as in that vein. So what does that mean? Well, it means if somebody, in particular pharmaceutical companies, coming up with a new whizzy chemical, in other words, a new drug, or some new surgical treatment procedure or whatever, then we always want to assume that it's going to be potentially harmful unless we can assert with very strong evidence that it's actually innocent. And then because of that, we have this very sort of strong and stringent system of clinical trials, phase 1, phase 2, phase 3, to make sure that it's working as it should work and not doing harm. And then we follow that up with real-world studies with particular regard to side effects. And as many of you will be reading about over the last few months, obviously the pace of the vaccine development has been quite extraordinary, and one of the key things following on from that, therefore, is that everyone's been very interested in the real world studies, because the vaccine clinical trials themselves are very much around efficacy in the context of mortality, with a fairly sort of well-defined, well, of course, age ranges, but the real world studies, after vaccination got underway, have then been very, very informative in thinking through what are the other things we can learn about the vaccines, such as impact on transmissibility, and also what happens in age ranges beyond what was looked at, and also it helps us to think through what's actually happening in the real world as opposed to the clinical trial settings, because as many of you may be aware, in the context of clinical trials, things are by definition fairly well controlled, in particular what sort of people you're starting with, and the subset of people who enter the clinical trial are somewhat different from the subset, or the set of people in the real world, which is what you're ultimately concerned with. But going off for a bit of a tangent there, but the point there is we've got a very strong procedure for testing things with the assumption that new things should be assumed guilty until proven innocent. And of course, thinking through that, one of the great wins for any pharmaceutical company, which can be a win for society as a whole, is this idea of repurposing. In other words, if we take one particular drug and we know it's been through all the trials 10 or 15 or whatever years ago in a different context, we can then say, well, we've proved that's safe, and we can then see if it's going to work in a new context without having to be quite so concerned about reproving this is not harmful. And that's actually been lots of repurposing in the context of psychiatric medicines, for instance. But of course, how all this works depends entirely on your starting point. Are you starting with the assumption that we need to assume things are guilty until proven innocent, or not? And things can be slightly different in different contexts. So if you look at this 1992 Rio Declaration on Environmental Policy, it's very interesting that it takes almost a different approach here, where there are threats of serious irreversible damage. The lack of full scientific certainty should not be used as a reason for postponing measures, which is a useful perspective to bear in mind, because it's simply saying if we wait years and years and years to accumulate evidence, it's probably going to be too late. And that's very much the situation I think we're in the pandemic. If you wait too long, then things will have progressed too far, and the status quo isn't normality. The status quo is we're in the middle of a raging pandemic with lots of people dying or being at risk of dying in coming months. So with that as a starting point, if we move on a bit, please, Dan. What I want to do in the next few slides is just think through how this applies in various different contexts. So face masks, and then the 1 or 2 days if like debate about the vaccination program, which is hopefully no longer a debate, and then also the aspect of blood clots, which is still a bit of a debate. So those 3 different decision-making examples hopefully shed a bit of light on all this, and I think they're very interesting in their own rights. So with face masks, on the table on the right, you can see the way in which different countries introduced face masks as a mandatory step in different months last year. So in March, Venezuela, Czechoslovakia, also the Czech Republic, also Slovakia, I think that month. Italy adopted a regional procedure. So Lombardia, where things kicked off, was one of the first to introduce face masks, as you might expect, in April. Many other countries may, and in England, for those of us in the UK or England, as we'll realise that England was one of the last to adopt these, with June for public transport, July for shops. I think some of the other regions of England, of the UK, were slightly earlier. There's always surprised many people, myself included, that England was so sort of slow to adopt this, which then sort of makes you think through what was the actual thought process behind that, both in England and also in the other countries. And if you sort of think through what I was saying in the previous slide or two, it all depends on this starting assumptions, whether you assume some new treatment, some new procedure is going to be potentially harmful until you've proven it's fine, or the other way around. So in other words, we've got 2 thought processes here. You could be thinking through, I'll assume face masks may be beneficial. Unclear who benefits most, but between the person wearing the face mask or the person that person might be sneezing on or coughing on, but that goes beyond the remit of this slide. And we might assume there's no serious question of harm, because unless you sort of swallow your face mask, you're probably not going to do great harm to yourself. So in other words, you might start with the assumption of innocence until proven guilty. And the other approach is more the one we've seen if we're sort of thinking through what happens if we have a sort of new drug such as a statin or something, and assuming it's guilty until proven innocent. And of course, the problem then is that you have a very, very long time to amass the evidence to prove they're worthy. And the problem in that context, of course, is that we're in an emergency, and just as through Dan's slides, we're sort of noting that the great advances made in data analysis in the context of the emergency. Can we also be more flexible in our thought process in the context of an emergency, where we don't really have time to wait months and months or years and years amass the evidence. And as I think Dan was implying at the start of his part of the talk, traditional epidemiology has been a sort of many years worth of data collection in normal times to sort of amass the evidence to make a decision. So a piece in the BMJ, the British Medical Journal, in early April summarised at the time the known evidence on face masks, which was actually, in terms of, if you like, traditional strong epidemiology, was a very small amount of evidence, which at the time that they sort of still regarded as being, the doctors writing this, I think it's Tricia Greenlaw, still recommended it being a strong case for face masks, you know, albeit it's not the sort of evidence you might be as conclusive about as you would be in other contexts. But again, it very much all depends on your starting position. Do you want to start with an assumption of innocence until proven guilty, or the other way around, which just goes again to the previous point about analysis not being analysis unless you actually think through what is your starting assumption, and is that completely valid. One of the things that me and Dan wrote about in an article last year for the longevity bulletin, the Institute and Faculty of Actuaries longevity bulletin about epidemiology in more normal contexts, and we've got a reference to this at the end of this section, is the Bradford Hill criteria. So when epidemiology was really getting going in the 60s and 70s, one of the prime epidemiologists, Bradford Hill, set out his criteria for sort of thinking through the results of any study, with particular regard to trying to ascertain causality, which is of course a key thing that epidemiologists are trying to ascertain, is something causal or not. And one of the things he noted was this aspect, which is completely vital, I think, of biological plausibility, which in other words is another form of evidence. So evidence isn't just the stats you're accumulating, it's also whether something makes sense in an overall biological perspective. And here, of course, with face masks, we can say, well, if we have this virus which is being transmitted through this respiratory mechanism, then another form of evidence is this the whole biological mechanistic aspect of, would we regard a face mask as being probably useful or not, just from the mechanics. And again, it just goes to show that epidemiology is more than just a question of data analysis, a question also of amassing different types of evidence and making a broader conclusion based on that. So it's a quick, or hopefully a sufficiently quick talk through the vaccine, the face masks. So on to vaccine, so 1 dose or 2 dose, and this has been a very interesting thing. I think if anything, the lesson, I don't know if it's been officially acknowledged or not, about the face mask aspect in England, Public Health England was very slow and probably completely incorrect decision making. Whereas with the 1 dose or 2 dose thing, things have been really much more impressive and much more flexible. So all the mainstream vaccines have been based on a two-dose strategy, and some of them are being talked about now with a 1 dose basis, which obviously would be very helpful. So that 2 dose strategy was tested as being 3 weeks apart, and when the way the clinical trials were devised, and that 3 weeks apart wasn't so much because the chemicals involved, if you like, physically required a 3 weeks of separation. It's more just the way the trials were devised. So it wasn't a sacrosanct figure. Just before Christmas last December, Tony Blair suggested that we move to vaccinate as many people as possible, and do so by having a 1 dose strategy, as opposed to vaccinating half the number of people with 2 doses. And at the time, I was partly because it was Tony Blair suggesting, I thought it was probably a load of rubbish until I started thinking through the numbers involved. But it's interesting thinking through some of the other reactions there. So the first 2 references from the BBC news article with that first link there. So 2 people being fairly negative about it. It's not entirely clear if those quotations are slightly out of context, but again, the overall tone is rather negative. The next 2 quotes from some sort of more news accumulated very soon after us by this Science Media Centre. One of them generally positive, Professor Openshaw, and one fairly neutral. But then the JCVI, the Joint Committee on Vaccination and Immunisation, sort of thought it through and gave a go-ahead to that 1 dose approach at the end of December, which is a very impressive sort of speed of response. And the next slide sort of talks through what our own calculations were on the subject. When I say our own, meaning the COVID-19 Actuaries Response Group. Dan, if you want to hit the button for the next slide, please. See the mouse moving around. Oh, there we go, thanks. So the reference to our posting here is the one at the bottom, which also gives them the subsidiary references to some of the things referred to here. What we did here was think through at a very simple level, 1 dose or 2 days, what can we do to shed some light on the question. We looked just at the Pfizer vaccine, but we could have done very similar calculations for the AstraZeneca. And with the Pfizer, we had the initial trial results was showing just 52% efficacy for people with 1 dose, but that was somewhat misleading because many of the trial volunteers would have been thought to have had COVID pre-vaccination. So the JCVI themselves had adjusted that to a range of plausible figures, of which 74% was at the lower end of the range. So the real figure is probably actually slightly higher. Probably be interesting to go and revisit all these now that we've got more data. But the comparison points therefore was we were assuming 74% efficacy after just 1 dose, or 95% after 2 doses. We could therefore take a group of people and work out what happens if you adopt either the 1 dose or the 2 dose strategy. And bearing in mind when we talk about 1 dose, we're not saying 1 dose and that's it. We're saying 1 dose and then get the second dose whenever when you can, which in the UK is typically a 12-week thing, as opposed to just 1 dose. So we take 1,000 people, 1,000 doses, and work out what's the optimal way of deciding on your approach. You could either have a 2-dose approach, you've got 500 vaccinated, 500 not, and you can see there we can split into at risk of severe COVID or not at risk. So if supposing you go for the 2-dose approach, you've got 500 vaccinated. If you go over to the right-hand side, you can work out there if you've got 475 not at risk because you've got the 95%. If you like, success on the 500 vaccinated, but everybody else is at risk. So at risk you got the remaining 25 plus the 500 unvaccinated. Or on the other hand, you could have the 1-dose strategy for the 1,000, and that gives you, again, assuming that 74%, gives you 260 remain at risk, 740 look to be protected. So again, the maths done in a fairly simple back of envelope way is fairly clear. And I think everyone was impressed by the sort of speed of the JCVI's thought process. And again, Britain moving into, well, certainly England and Wales, I think the other territories moved in a similar way. That speed of response, I think, impressed many people and gave us a great, gave us, meaning Britain or the UK, a great edge in the vaccination, well, effectively a race. So that's that on that one. And vaccines and blood clots, again, the most recent debate, and this will just be last minute or so, and then we can move on to more Q&A time. There was, of course, widespread concern around increased risk of blood clots from the AstraZeneca vaccine. But the key issue here is, again, almost goes back to that starting point with the interpreting the results from your epidemiological data, interpreting the results of your analysis. What are you actually comparing against? So when many of our like some European sort of friends and partners, as Boris Johnson would probably refer to them, started slamming the AstraZeneca vaccine because of the blood clots. Again, they're effectively making it a comparison of the risk of a blood clot from AstraZeneca compared with the risk of not taking anything in a perfect world, absent COVID-19. But of course, the comparison we really have to make isn't that comparison against the ideal world with no COVID-19, it's a comparison against the world in which we currently are with a raging pandemic. So in other words, we then have the comparison needed of the risk of blood clot from the AZ vaccine against the risk of blood clots from COVID infection. And that graphic on the bottom left shows also some other sort of interesting comparison points. For instance, the risk of blood clots against birth control pills. And of course, society has never been particularly averse to birth control pills on the grounds of massive blood clot risks. Or with smoking, it's never been particularly concerned about blood clot risk from smoking as opposed to all the other nasty ways it can kill you. So that helps put things in perspective. On the right, from the Winton Centre, it's a useful graphic, which then also relates to the reason why I think the UK has taken these sort of age decisions it has regarding the AZ vaccine, that if you're in that youngest age group, then the blood clot risk is sort of material compared with the related COVID risk, so sort of less advisable, whereas for the older age groups, you can see there the 50 plus, 60 plus, the blood clot risk from COVID against AZ is just almost no comparison. So again, hopefully that's a useful thing for sort of thinking through the thought process there and making it clear that, again, with any epidemiological results is all around what you're making your comparison against, and in the case of the pandemic, we don't actually have an ideal world to compare against. So a quick canter therefore through some aspects which hopefully shed a bit of light on the thought process and how that thought process may need to change in the world in which we are at the moment. And there's just a final slide, just putting in a couple of links. One is an article that Dan and I did in the Longevity Bulletin on our epidemiology, so if you like, in pre-emergency times. And the second link is the presentation we did on that, pretty much the same subject. And the bottom link, obviously, sort of advertising a bit our own COVID active response group, where we've both written various articles on different things. So that should be it, really. So have you got 10 minutes or so for Q&A if people are busy typing in things?
So Matthew, there's one question that's come through already, anonymously, and that relates to what you were saying on face masks. And as we go forward, the question is, how long do you believe that people will continue in different countries to kind of continue to use face masks? And are we seeing a new normal similar to that which we have seen in various different Asian countries in the future of widespread use of face masks?
Yes, very interesting question. In fact, when I was preparing the slide for the talk, one of the most recent sort of questions being debated in the British Medical Journal, I think it was only last week actually, was whether we should be starting to think about face masks in open air settings, as well as indoor settings. But that's again in the current time where there's still non-trivial risks involved. So I mean, to my mind, all of these non-pharmaceutical interventions and other control measures, which are, well, as you might guess from the word control, to a large extent restrictive, are there to cope with a problem. And if the problem is largely gone, or in many places such as New Zealand completely gone, touch wood, then it makes sense for those measures to be relaxed. And again, individuals can always decide to wear face masks or lock themselves away if they wish to. But it seems to me that from a societal point of view, it's beneficial to normality in societies to minimize these measures as far as possible. And I suppose there may well be a halfway house. I can well imagine if there's a winter flu resurgences, as there probably will be from next winter onwards, that people may get back in the habit of wearing masks in crowded indoor places or on public transport, rather like they tend to do in many countries in Asia, without it necessarily being mandatory. But it's very much seems to be a political and societal perspective there, which will vary, in fact, I think, a lot from country to country and also from age group to age group.
So another question that's come in a little bit earlier on related to variants, which I'll take if I may, and that's looking at to what extent variants that have been detected, where there are particular variants of concern that were detected in a kind of high sequence areas, and as a result of that have been either eradicated or restricted. And I guess that there are two interesting pieces to this. The first is, would we be able to say that it was through our actions that actually we prevented a particular variant in the early course of the pandemic? And the second relates to more recent experience that we have, where there's a relative flooding of areas with testing because a particular variant has been detected. I think certainly in the early phases, it would be very difficult to tell the difference between our ability to restrict and the variant's ability, or a new variant's ability, to outperform the existent variant. And going back to one of the earlier slides, you could see there was a whole period where there are a whole number of different variants that actually have since disappeared in numbers, and that's probably throughout competition. But that is out competition during a period when we probably were making things more difficult for the virus because of restrictions. Certainly with the greater focus and the greater levels of sequencing, it may be possible going forward to say whether, for example, we've been able to hold back the level of a particular variant, either the South African or Brazilian in particular countries. But so far, for some of those earlier variants, I don't think we have the information to say that it was because of a lockdown rather than because of an out competition. Another question that's come through, maybe Matthew, you want to start with that one, which is just in terms of the one from Louise talking about what epidemiologists have learned that will affect how epidemiology is done in the future, new techniques for analysis or data collection?
A very tricky one, isn't it? I mean, because at the moment, I think again, going back to that 1 days versus 2 days thing, where I think we have been impressively sort of mentally flexible compared to what you might have imagined would have happened 5 or 10 years ago in normal times. It's really just been a question of being in crisis mode and needing to respond, bearing in mind that we're in crisis mode. I don't know whether the actual decision making in future will reflect that, or I mean, cause I think decision making needs to reflect the situation you're in. Whereas maybe Louise's question more around the technology behind the data collection, which strikes me a lot of the data collection is being done. The EHR, the electronic health record stuff, has been on the one hand very strong, as seen in that open safely study, although obviously not as strong as it might have been otherwise. It would have been updated. But all the information collected on smartphones is presumably is quite prone to data bias, and one of the hidden problems with epidemiological studies is data bias, which again in times of crisis as we are now, it's still been, as you were saying, Dan, immensely useful, but you sort of wonder to what extent some of these things can be used with massive confidence. But I mean, I'm sure you could add more insight than I could on that one.
Yes, no, I mean, I think you covered that very well, but I definitely believe that we have seen over the last year and a half two very significant shifts. One is a much wider group of scientists and data experts looking at epidemiological problems than historically has been the case. And the second is obviously the enormous amount of data that's being gathered that previously wasn't gathered outside of a very controlled study. I mean, there's a related question which came a little bit earlier, which I quite like because it kind of takes me back to the very beginning of
it's a little bit
the
like
presentation.
A cholera-based question, isn't it?
Yes, indeed. I mean, you know, the anonymized sampling of population information is exactly where John Snow started. You know, he a priori was looking at these two water companies and trying to compare who effectively was pumping sewage basically into people's houses, not to put too fine a point on it, compared to those that were drawing the water from slightly higher up, and was then looking at who developed cholera. And I think that anonymized sampling of population data and getting local information is effectively what we're seeing more and more, where for example, a variant is sequenced, and that variant immediately leads to kind of canvas lateral flow tests and PCR tests in that region, and also kind of a particular person is diagnosed on a plane, and then we look at secondary contexts. So the ability to localize that information and to gather a much greater level of information than would be necessarily available from a verbally provided survey, then allows kind of machine learning and allows greater insight. So yes, I think that we are seeing a shift from the planned epidemiological study to the type of Zoe information, which is based on a huge amount of data being collected and then analyzed after the event. Matthew, do you want to take the...
Yes, probably it's a good question, and probably the last question we have time for, but in terms of data gathering, e.g. ONS sampling, how significant is dishonesty or false reporting in general, which I think has always been a big one. So I think back in the summer of last year, we were sort of within the COVID-19 action response group, we're having quite a lot of discussions around some of these samples, the surveys around mask wearing, because in fact we're sort of looking at the results being published and comparing that with what we were seeing ourselves anecdotally in all the different parts of the world we live in. It always seemed to us that the mask wearing was being exaggerated by a factor of 5 or 10. You know, people might be sort of, well, we were seeing 50, I think 50% reporting mask wearing, where we were sort of thinking it's more like 5 or 10% at most. So I think there's a huge problem there. Again, it depends very much on what decisions are being made on the basis of that, and also whether the interpretations are being made allowing for that dishonesty. This is very much an open question, as it is with any survey, rather like all the political sort of polling problems, where sort of people are generally sort of maybe embarrassed to they vote for particular parties, and such parties then generally do much better than the polls predict. Yeah, that's probably the last question we have time for, so thanks very much for all that. I think when the RPO is going to be very efficient, and send out the sort of link to the presentation and the copy of the slides very soon. And on that final slide, we can add mine and Dan's contact details if there are any further questions. But thanks very much to all of you for either having dialed in or watching this if you watch the recording in many years' time from your post-pandemic bunker.
Well, thank you very much, everyone, and wish you a good rest of your day.